let's finish out the chapter, the cytoskeleton. This is what maintains the cell's shape. Just like your skeleton determines your shape, the skeleton of the cell determines what shape it is. The cytoskeleton also assists in the movement of things within the cell, including organelles, and it makes internal transport possible. So how do those vesicles get from the ER to the Golgi body to the cell membrane? And that is done via the cytoskeleton. It's like a road for those vesicles to travel on. There are 3 types of fibers that make up the cytoskeleton. They are actin filaments, intermediate filaments, and microtubules. And these can be assembled and broken down as needed. And they can move. They can cause the cell to move or change its shape depending on what the environment needs. Here we have pictures of the fibers in different organisms. So here we have some filaments of a plant. Here we have the intermediate filaments of a peacock. And here we have the microtubules that are found in a chameleon. Actin filaments are extremely thin filaments that are twisted like a pearl necklace. They form a dense web just under the plasma membrane and maintain the cell's shape. Microvilli are little extensions of the cell, kind of like cilia, that are really good at absorbing things. So they are found in your intestines. That's how your nutrients get absorbed. And in your lungs. That's how gases are exchanged. So these actin filaments provide support for these microvilli. The actin filaments are also used for intracellular traffic control. So they're like the roads for how things are transported from one place to another. And they are responsible for cytoplasmic streaming, which is the cycling of all of the organelles in the cytoplasm. They just kind of flow around. The actin filaments are also responsible for the pseudopod amoeba movement. So amoebas are single-celled organisms that live in water, and they will actually change the shape of their cell. They will create this appendage that looks like a foot, and it will stretch as far as it can, and then the rest of the cell will kind of like slingshot to join it. It's pretty cool movement. I will link a video on Google Classroom if you are interested in seeing that. Actin filaments are also important in muscle contraction and important in animal cell division. That's whenever we take one cell and divide it into 2 cells that are identical. So here we have our actin filament. All right, looks like that twisted strand of pearls, and it is connected to the membrane by myosin, and they use ATP, and the actin filament will stretch this way, and it will cause the membrane to shift as well. Intermediate filaments, they're called intermediate because they're between the size of actin filaments and microtubules. And they have a rope-like assembly of polypeptides, so they're protein. They vary in nature from tissue to tissue, so depending on what type of cell we're looking at. And their functions are to support the nuclear envelope and create cell-to-cell junctions. Some cells have to work very closely with their neighbor. So skin cells are a type of cell that has to do that. So the intermediate filaments form cell-to-cell junctions that hold those skins together. They also provide the protein keratin. They're made out of the protein keratin, and it gives them a lot of their strength. Microtubules are hollow cylinders made of 2 proteins called alpha and beta tubulin. Spontaneous pairing of alpha and beta tubulin molecules form structures called dimers, di meaning 2, and they arrange themselves into spirals, and each of those spirals has 13 dimers. They are assembled under the control of the microtubule organizing center, or MTOC, and these, the most important of these is the centrosome, which is used in cell division. It interacts with motor molecules, kinesin and dynein, and causes the movement of organelles. Because sometimes an organelle isn't in the most efficient place and it needs to be shifted a little bit. And so these kinesin and dynein molecules help move them. They also help with cell division. The mitotic spindle is what causes the chromosomes to be divided evenly whenever the cell needs to divide into two. So the mitotic spindle is made of microtubules. And we're going to talk more about that later this year. Here is microtubule operation. Okay, so we have a vesicle, and it will join to a kinesin receptor. Here we have kinesin, and it will attach itself to the microtubule. And it basically just walks its way down the microtubule. The vesicle moves, the microtubule does not. Just like a car moves on the road doesn't move for the car. Centrioles are short hollow cylinders composed of 27 microtubules that are arranged in 9 overlapping triplets. There's 1 pair per animal cell. They're not found in plant cells. And they are oriented at right angles to each other, and they help during mitosis to make sure that everything is divided equally. And these centrioles also give rise to the connection point of cilia and flagella, which you will remember are the structures that are responsible for cell movement. Here is the centriole structure. All right, so you can see they are in triplets, 1, 2, 3, and there are 9 of them, and they form a spiral shape. I always think they look kind of like churros myself, but you know, I'm very food oriented. Cilia and flagella. Cilia are hair-like projections from the surface of a cell that help it move. All right, in eukaryotic cells, the cilia and flagella are very different. They have an outer covering of the plasma membrane, and inside there's a cylinder of 18 microtubules arranged in 19 pairs. And in the center there are 2 single microtubules, so this gives us a 9+2 pattern that is used by these extensions of the cell membrane. In eukaryotes, cilia are much, much shorter than flagella. I like to compare it to like jazz hands are cilia, and flagella are more like a whip. Cilia move in coordinated waves like oars, and they're used in your digestive system to absorb nutrients and in your respiratory system to facilitate gas exchange. Flagella are more like a propeller or a corkscrew, and they're found in sperm cells. Here is the structure of the flagella. Here's our 2 in the middle, surrounded by our 9. All right. So there is the structure. And that's it. If you have any questions, let me know.